Good morning, my dear friends. Myself, Dr. Abhishek, working as a professor in the Department of Pharmaceutics. Today, I am going to explain about the topic application of recombinant DNA technology. In our last class, we have already discussed about recombinant DNA technology. In brief, we can understand here we are having the DNA which we have received from any donor cell and for which we are going to multiply. That is the DNA, and this DNA will attach into the plasmid by the attachment of the plasmid, and it will work as a carrier. So this plasmid will introduce into host cell. This plasmid will introduce into the host cell. So here the host cell may be E. coli. So in this host cell, it will get sufficient protein synthesis. By the help of this protein synthesis, this multiplication of this DNA can be possible. So this was the brief introduction of recombinant DNA technology. Today we will discuss about some important applications of recombinant DNA technology. So the first application is molecular biology. We are using the technique of recombinant DNA technology into the molecular biology. So how we are using into the molecular biology? There is one method, that name is gene mapping. So what do you mean by gene mapping? Gene, you know very well that DNA which we are going to multiply, but mapping means identification. How to identify, how to recognize. This is the genome. From this genome, this is very difficult to identify our target gene. But from this genome, we can, by the help of gene mapping, we can recognize this target gene. Where is the location of this target gene? We can recognize it, and by the help of restriction endonuclease, we can cut this gene and we can make it separate, which is going to amplification. We can cut this gene from the genome and we can make it separate, and for this gene we can, after that this gene we can use for recombinant DNA technology or for the multiplication. So this is the gene mapping. This is very, very important technique. The next technique is genetic disorder. Into the genetic disorder, how recombinant DNA technology will work into the genetic disorder. We know very well many type of disorders are there, many type of defected gene are there, into that defected gene, how we can mark to the defected gene. So here we are having a DNA probe. So by the help of this DNA probe and fluorescent dye, by the help of fluorescent dye, we will dip this DNA probe into the fluorescent dye, and we can, by the help of that probe, we can mark to the defected gene, and by the help of this technique, we can recognize or we can identify our defected gene, and the problem is arising into the genes, and that can be treatable. After recognizing or after identification, we can treat it well. The next technique, the third technique, is monoclonal antibodies. the production of monoclonal antibodies. This is the third application which we are using into the recombinant DNA technology. So from very basic we will understand here what do you mean by antibodies. What is antibodies? See, as any infection or any germs will attack onto the body, body will start the formation of antibodies. As the infection will attack to the body will start formation of antibodies. So antibodies will fight against the infection and protect to the body. Antibodies will fight against to the infection and will protect to the body. This is the benefit, this is the significance of the antibodies. So antibodies is very useful product of our body. Our body will produce antibodies, but monoclonal antibodies, what is monoclonal antibody? Monoclonal antibodies prepared in the laboratory. Monoclonal antibodies will prepare into the laboratory, and by the help of this monoclonal antibodies we can protect the body from any dangerous infection. Have you seen about the coronavirus or some other virus that may be the cancer virus, AIDS virus? So from this monoclonal antibodies we can protect our body. How it will protect? You know very well, this is the infectioning agent and this is the surface of the body. As it will introduce into the body, as this infection will enter into the body. So what this monoclonal antibodies will work? Monoclonal antibodies will form a covering surround this microorganism. These monoclonal antibodies will form a covering for this pathogen. By the help of this covering, it will make deactivate to this microorganism. This germs or this pathogen will not show any harmful effect onto the body. By the help of this covering, it will use to destroy this microorganism. So this is the third important application, that is production of monoclonal antibody. Now the next application is gene therapy. So how we are using recombinant DNA technology into the gene therapy. From the therapy word you can understand, by the help of therapy word you can understand gene therapy. So here we are going to treat, or we are going to change the faulty DNA, the defected DNA. If in the DNA we are finding any defect, this is the defected DNA. So this defected DNA we can replace by the happy DNA. Happy DNA, or you can say healthy DNA. Healthy DNA. So this faulty DNA we can replace by the healthy DNA. That you know very well the technique of the recombinant DNA technology. So into the recombinant DNA technology, this technology we will apply here, as we can cut this faulty DNA by the help of restriction endonuclease. From the restriction endonuclease, or we can by the help of molecular marker we can mark it here first, and then after that we can cut this portion which is defected, and this fresh portion we can replace or we can attach here. See, restriction endonuclease will work at the time of we are going to cut our faulty portion from the DNA. We will use restriction endonuclease, and DNA ligase we will use when this happy DNA is going to attach into the faulty DNA. This DNA ligase we will use it here, and then we can replace the problem. We can replace our defected gene to the happy gene. In this way, we can use recombinant DNA technology in our gene therapy. The next one is production of vaccine. The next one is production of vaccine. Production of vaccine. The next one is production of vaccine. What is vaccine? Vaccine is a substance. Vaccine is a substance which is used to protect or which is used for the immunity of our body. Vaccine is the substance, but how we will how we are going to prepare the vaccine? How we can prepare the vaccine? For the vaccine, we will. This is the pathogen. This is the pathogen. So here, for the production of vaccine, we will use a portion of the pathogen. Portion of pathogen. We will use a portion. This portion we will use. We will cut this portion and we will use this portion for the formation of our vaccine. So this is the, what is this portion? Protein. We will use protein for the formation of vaccine. We will cut this protein and we will take, we can inject or infuse. By the help of injection or by the help of infusion, we can transfer this vaccine into the body. We can transfer it into the body, and what will happen when you are going to inject this protein into the body? So body will recognize, body will recognize this protein. So by the help of this portion, by the help of this protein, body will recognize it and start producing antibodies. It will start producing antibodies. So as per this microorganism, as per the nature of this microorganism, as per the nature of this pathogen, our body will respond and it will start formation of antibodies. So sometime if we are not going to give vaccine into the body, you can see in the case of any viral infection or any coronavirus infection, if a person is infected from any disease, so antibodies will develop into the body without any vaccination and all. So this is also a type of immunity. The next one, the next application is DNA fingerprinting. So what is DNA fingerprinting? From by the name of fingerprinting, you can understand recombinant DNA technology is the study which we can use in the fingerprinting or recognition and identification. Here we can identify by the help of DNA fingerprinting, we can find out the DNA of any criminal or any crime scene. Nowadays into the crime scene we are using this technology, DNA fingerprinting, and by the help of this DNA fingerprinting we can differentiate the gene of criminal, and the gene of victim. So in the crime scene, you can find out the both of the DNA of the criminal and the DNA of the victim. So by the help of this technique, by the help of this DNA fingerprinting, you can easily differentiate between the DNA of criminal and DNA of victim. From this DNA, by the help of DNA fingerprinting and by the help of recombinant DNA technology, we can recognize the parent cell of any daughter cell. We can recognize the parenting of any newborn baby. The next one is the production of pharmaceutical product. Pharmaceutical product, nowadays we are using many pharmaceutical product which are coming under pharmaceutical biotechnological industries. So by the help of recombinant DNA technology, we can start our production of biotechnological product. As you know very well, insulin is very much useful. Insulin is a product of the body only. Insulin is the product of body, but we require insulin in case of diabetes. So how we can go for the production of insulin? This is the genome. From this genome, we will recognize the gene of insulin, and we can cut it. We can cut this gene and we can go for the multiplication. We can start multiplication by the help of plasmid and by the help of host cell. By the help of plasmid and by the help of host cell, we can go for the multiplication, and this is known as type of cloning. From this technique only, by the help of recombinant DNA technology, we can go for the production of many pharmaceutical or biotechnological product. So thank you so much, guys. These are the some application which I have explained. These are the application of recombinant DNA technology. Recombinant DNA technology is very useful. So by the help of this class, I cannot complete all the applications here, but few of some important application I have explained. By the help of these explanation or by the help of these application, you can explain it well in your examination point of view. This is very important topic. Thank you so much, guys.